Today we're going to talk about the microscope in cell studies. Here are your Cambridge objectives for understanding about the microscope. So you will use an eyepiece reticulum stage micrometer in the lab once we get into lab. But for now, you're just going to learn how to use them. Okay, so the common units of length that are used in cell studies are millimeter, micrometer, and nanometer. So measuring cells using a microscope requires cells and organelles that can be measured with a microscope by means of an eyepiece reticule. This is a transparent scale. It usually has 100 divisions. The eyepiece reticule is placed in the microscope eyepiece so that it can be seen at the same time as the object to be measured. Usually it's already in the microscope when you use them. Not all microscopes have them in the eyepiece, but most do. And we will not know the actual size of the eyepiece until the eyepiece reticula is calibrated. So it's kind of like a random scale. To calibrate the eyepiece reticule scale, a miniature transparent ruler called a stage micrometer is used. The scale is placed on the microscope stage and is brought into focus. So this scale might be etched onto a glass slide or printed onto a transparent film. All of the ones I've ever used are a glass slide that you either put on top of the piece that you're looking at or just straight onto the stage of the microscope. It commonly has subdivisions of 0.1 and 0.0 millimeters. So we're going to talk about the differences between resolution and magnification with reference to light microscopy and electron microscopy. So resolution is the ability to distinguish between two objects that are very close together. The higher the resolution of an image, the greater the detail that can be seen. Whereas magnification is the number of times larger an image is than the real size of the object. And the equation that you need to know for that is the observed image size over the actual size. So the best way to do that is to have a triangle with image over magnification times actual size. Okay, let's talk about light microscopes. Microscopes that use light and optical lenses to magnify tiny objects are called light microscopes. Sometimes they're referred to as optical microscopes. Light microscopes have been used for more than 400 years and are still really the most common type of microscope in use today. In the 1600s, a Dutch biologist developed a really simple optical microscope, which only had one lens and used the sun as a light source. So compound microscopes and light microscopes with more than one lens had been invented in the 1500s, but really could only magnify to about 40 times. And the Dutch biologist magnified to over 200 times. He became the first scientist to see tiny single-celled organisms living in water, on his teeth, and his bloodstream. It was really, really awesome. And he called them animalcules, which today we know to be protozoa, bacteria, blood cells, etc. So later, compound microscopes became more sophisticated and can now magnify objects way more than simple microscopes. Almost all light microscopes used today are compound microscopes. Most modern compound light microscopes have the same basic components. The first is an eyepiece containing an ocular lens. This is the part of the microscope that you look into, and it provides some magnification of the object you are looking at. And typical magnifications of the ocular lens are about 2 times to 50 times. They also have an objective lens, which is placed just above the object you want to magnify and magnifies the object even more. And then what's really important is a light source, which illuminates the object, and a movable stage holds the sample and can be moved up and down using focus knobs to bring the image into focus. So the light, really what it's doing is providing light rays that can pass through the specimen on a side and are focused by an objective lens and an eyepiece lens. So this produces a magnifying image of the specimen on the retina of your eye. Alternatively, the image can be projected onto a screen or recorded by a camera. Generally, the magnification possible is times 1500. So here are some examples of light or compound light microscope images. Okay, electron microscopes. So optical or light microscopes use light to create images, but there is another type of microscope called the electron microscope, which uses a beam of electrons to image tiny objects and structures. So because the wavelength of an electron is much shorter than the wavelength of visible light waves, electron microscopes can achieve much higher magnifications that can't be seen even with an optical microscope. Electron microscopes are commonly used in the study of viruses, viral diagnoses, which are too small to study with another type of microscope. So really there are two types of electron microscopes: transmission electron microscopes, or TEM, and scanning electron microscopes, or SEM. So first let's talk about a transmission electron microscope. So TEM passes a beam of electrons through a very thin section of a specimen. The interaction of the electrons with the specimen is detected and used to create an image. Transmission electron microscopes have a very high resolution and can even allow you to see single atoms. This would never be possible with a light microscope. Instead of passing a beam of electrons through the specimen, it passes the beam of electrons through the specimen. The final image is two-dimensional. And the magnification possible with the TEM is times 500,000. Here are some images created by electron microscopes. Okay, so for scanning electron microscopes, instead of passing a beam of electrons through a specimen, a scanning electron microscope passes a beam of electrons over the surface of an object to create a three-dimensional image. Although scanning electron microscopy does not have as much resolution as transmission, it can still image objects smaller than 1 nanometer. In addition, because scanning electron microscopy creates a 3D image, it can show surface structure better than any other type of microscope. The magnification possible with an SEM is about times 100,000. So here's just a table summarizing the resolution and magnification specs for light microscope, transmission electron microscopes, and scanning electron microscopes. So looking at animal and plant cells, every plant and animal cell is surrounded by a thin cell surface membrane. All have a nucleus, which is very large and stains darkly. And the nucleus located within, the nucleolus, located within the nucleus stains even more deeply. So those are those even darker purple spots within the purple nucleus. The material between the nucleus and cell surface membrane is called the cytoplasm, and it's the fluid part. And technically, anything that is outside of the nucleus is considered the cytoplasm. Many small structures called organelles are seen within the cytoplasm. Organelles are functionally and structurally distinct parts of a cell and are often surrounded by membranes so that their activities can be separated from the cytoplasm. This is called compartmentalization, which is really a division of labor, a sharing of work between specialized organelles within a cell. The most numerous organelles seen with light microscopes are usually mitochondria. Golgi apparatus can only be seen with silver-containing stains. So only in animal cells will you see a centriole. This is involved in nuclear division and appears as a small structure close to the nucleus. And only in plant cells are you going to see cells that are surrounded by a cell wall and linked to neighboring cells by plasmodesmata. Let's take a minute, go ahead and pause the video and take a minute to really look at that image. Also, only in a plant cell, you're going to see a very large central vacuole, which is surrounded by the tonoplasts. You'll also only see chloroplasts in plant cells. Okay, so we need to be able to calculate actual sizes of specimens from drawings as well as the linear magnifications from drawings. We are going to do this in class. So we will be doing labs as soon as labs start up next week. And we're going to have in-class assignments and tasks on Thursday that we'll use to achieve these goals. So right now, though, I thank you for watching the video, but I would like you to navigate to Learning Catalytics and take the completion quiz. Thank you.